Hey, the purpose of this segment is following up from the last segment where we went through the process of transcription and translation with a special emphasis on interpreting DNA sequence data to predict what protein sequence would be encoded by it from bacterial systems. We are going to continue by doing an example where we look at a DNA sequence and we ask ourselves what protein sequence would that DNA sequence be able to encode. So on the whiteboard screen here, we're given an example problem similar to something you might be asked on an upcoming quiz, for example, and you'd want to have your codon table handy for that because I don't expect that anybody has that memorized. I definitely don't. So we're asked here to predict the possible protein sequences encoded by the DNA sequence below. So as we look at this DNA sequence, the thing that I advise you to do first in looking at a DNA sequence is we keep in mind that if we have just this one sequence written here, we assume that it's written in the 5' to 3' orientation. And we look through that sequence for start codons. So number one, look for start codons as our workflow here to predict the protein sequence based on this DNA nucleotide sequence. Going through here, we're looking for ATG because ATG becomes AUG at the level of the messenger RNA. That's why we have AUG on the table here. So looking through this sequence, looking for ATG, looking closely, do we see any ATGs? And the answer is we don't. So at this point, do we throw in the towel and we say, well, there are no open reading frames in this, there are no proteins encoded by this particular DNA sequence, or do we look in some other direction? Well, I probably wouldn't be doing this problem if there was absolutely no protein encoded by it, let's be real. So what we need to do is take another step in order to look for open reading frames. And that other step is keep in mind that DNA is a double helix. And so there is the strand that I've listed here, and there's also the complementary strand. And it could be the complementary strand where we find an open reading frame. And so we look for start codons, and as an addendum to that, keep in mind the start codon could be on the other strand. The start codon could be on the complementary strand, in other words, the one that is not yet shown. It is possible there's no start codon at all, but usually if I'm giving you a problem, for purposes of this class, there's going to be something to do there. So the complementary strand, remember, is written in the opposite direction. 5' at the right-hand end, 3' at the left-hand end. And filling this in, we list the complementary nucleotide in each case. So C is complementary in hydrogen bonds to G, A to T, G to C, T to A. I'm going to go all the way down the chain here, going through this process of listing the complementary nucleotide. So continuing onward here, copying down my complementary base pair, the hydrogen bonding partner of that first strand is what I'm listing here on our second strand. And now we take a look through that strand for open reading frame, specifically looking for a start codon because there has to be a start codon there in order to make this work. And we keep in mind that the start codon may be on the complementary strand, and that complementary strand, rather than reading from left to right, we're always reading 5' to 3', which in the case of the complementary strand that's on the bottom means we're reading from right to left. So the strand that's on the top, we read the standard direction from left to right to get 5' to 3'. On the other side, since this is the complementary base paired strand, it's going in the opposite direction, 5' on the right, 3' on the left. And so the orientation that we need to be reading for purposes of finding the open reading frame is we are reading in this direction from right to left there with my rather pathetic crooked arrow there. So looking for an ATG reading from right to left. Do we see anything here? Taking a look through this, looking for An ATG scanning through. Oh, right here we have one, ATG. Are there any other ATGs? We just want to make sure we're not missing anything, any possible start codons along the way. And indeed, I see that the only start codon here is our ATG. It is possible that we could have a DNA sequence that encodes for more than one possible protein. So it's important to look and find all of the possible start codons, not just one. Then in order for that start codon to work out, after you have found that start codon of ATG, reading from the top strand or the bottom strand, in this case we had to read from the bottom strand, then we have to look for a stop codon that is a multiple of 3 from our start codon. This is essential because this is going to form the end of the protein. So we look for a start codon that is a multiple of 3 from the start codon. If it's not a multiple of 3, that is what we call out of frame. And an out of frame stop codon is not going to be suitable for stopping the translation process. Those types of mutations can occur in cells where a nucleotide is deleted, and that can, so-called frame shift mutation, can result in all the codons being shifted over by something like 1 nucleotide or 2 nucleotides, and that will completely screw up the sequence of the protein. It can make the protein a lot longer than was intended, because adding a nucleotide and getting rid of that stop codon as a result, making the stop codon out of frame, in other words, would result in the protein transcription and translation process just continuing onward until stop codon was found. On the other hand, depending upon where the nucleotide were inserted or deleted, it could result in a stop codon coming into frame and making the protein much shorter than anticipated. And that's one of the ways that genetic mutations occur and can cause a variety of negative health consequences, is they create these mutations that result in defective proteins, proteins that are too long or too short and otherwise not functional any longer. And so there's a whole field of human genomics that is growing very rapidly that looks at exactly this type of thing, mutated DNA sequences leading to mutated proteins, and trying to link that to specific genetic diseases. So anyway, getting back to the point of our problem here, we found a start codon on that complementary strand, and now we need to look in increments of 3 for a stop codon that is in frame. So it has to be a multiple of 3 from the start codon, in other words. So I'm blocking off in increments of 3 here, drawing out with my green highlighter marker here. And we need to find a stop codon, and our 3 stop codons are shown in red in that table in the upper right here. So we're looking for TGA, TAA, or TAG, and continuing onward, looking for a stop codon. Oh, here we have TAA. Thank goodness that is a stop codon that we recognize. And so I'm going to highlight that TAA in red as our stop codon. And this particular peptide that we are creating here, a very simplified peptide, since it is only going to have about half a dozen amino acid residues, in contrast to real proteins that have way, way more than that. We can go ahead now and translate that. So I'm going to write that the translation, and then I'm going to fill that in with the amino acid sequence, listing from the N terminus to the C terminus. And I'm also going to go ahead and update my flow chart here, my list of things to do to complete this process. So we look for a start codon, we look for a stop codon that was a multiple of 3 from it, and now what we're going to do is write the amino acid sequence predicted by reading the codons, those blocks of 3 nucleotides, from the start to the stop codon in order. And since we're reading on this bottom line here, we are reading entirely from right to left. So our start codon, reading from right to left, ATG, is going to be methionine. So I'm going to fill in methionine at my N terminus. CCC, looking through our codon table here at the C's, CCC right here corresponds to proline. So I'm going to fill in either P or PRO. I'll use the 3-letter abbreviations here since that is what's included in the table. And then we come to the next piece of the puzzle, which is GTA. Remember, reading from right to left here all the way across since we're on this bottom strand. GTA is going to correspond as we come over to here. GUA is the RNA equivalent of GTA, and that corresponds to valine. So I'll fill in valine here. So we've got our first 3 amino acids in place. We come on over to codon number 4, which is GGG. I come on over to the table here, I find G, I link it to another G, and then GGG is glycine encoded there. So I'll fill in glycine, our only achiral amino acid, GLY, as the 3-letter abbreviation for that. So that was our 4th amino acid of the chain. So we took care of the first one as our start codon, 2nd, 3rd, 4th. I've put red check marks on all of them just to keep myself in line here and in order, not skipping any or listing any twice. And we come on over and we have ATA or AUA if we're reading from the perspective of messenger RNA. So AUA, A, U as the 2nd letter, AUA is going to be isoleucine, ILE. So I'm going to go ahead and fill that in. Isoleucine, ILE, and that is our 5th amino acid in the chain. And then finally we have TAA, which we come over to here, synonymous with UAA from the perspective of messenger RNA. So that is our stop codon. And so as a result, I can plug in here at the end that this is our C terminal, has isoleucine, the N terminal had methionine. So this is going to be a sequence of 5 amino acids in total because that last codon encodes just for the stop of the transcription and translation. It doesn't encode for a specific amino acid. So keep these pointers in mind as you're looking for codons and looking specifically for what amino acid sequence those codons will encode. You have to find a start codon and a stop codon that are a multiple of 3 from one another. And those start codons and stop codons can be found on either the top strand as listed, or you may have to provide the complementary strand. And if you do, you have to read that complementary strand from right to left in order to find that complete coding sequence. So we will get into this topic more as we are looking at genomics. We'll talk about some of the computational methods that are used to interpret genomics data to predict what proteins are encoded by the DNA sequence. We'll also, as we go through the semester, look at ways to manipulate these DNA sequences to enable us to study the chemistry of these biological systems. So stay tuned for that.